Hello, everybody. Thanks for joining us today. Welcome to all the new attendees here, and if you're returning, we're happy to have you back. Criterion Edge runs multiple informative webinars throughout the year. Today is a live question and answer session. We'll be discussing the comparison of critical elements of the Scientific Validity Report versus the Clinical Evaluation Report, State of the Art, and Best Practice Writing Strategies. Our first presenter today is Lori Mitchell, founder and president of Criterion Edge, a global medical and regulatory writing firm serving the medical device, pharmaceutical, and biotech industries. Lori has over 20 years of experience in medical writing, safety, and pharmacovigilance management and regulatory reporting. Having provided regulatory and medical writing solutions to many pharma and medical device companies, both large and small, she is a proven leader in designing practical strategies to meet current global regulatory challenges. Lori is a published author and holds a Master of Nursing from UCLA. Also joining us today is Dr. Sarah Chavez, the Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience, and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I will welcome Sarah to give an overview of our session today.
Thank you, Olivia. So in today's webinar, we want to keep this very interactive, but we wanted to begin by outlining the must-have elements for these two very similar regulatory reports, which are the state-of-the-art section within an MDR-CER, as well as the IVDR's Scientific Validity Report, or SVR. So by the end of today's Q&A session, we hope that you will all have a better understanding of the required content within each of these documents and the documentation of the process that the notified body expects to see, the importance of planning and scoping this project before you even begin to write the state-of-the-art section, as well as hopefully gain some practical strategies to support the successful completion of these writing projects. So as we get started, I wanted to refer everyone to one of our white papers that we've published on our website that really details the purpose behind the state-of-the-art section. But this graphic does a really good job of explaining how many different pieces this section, this document will feed into. It will inform your risk management through your risk assessment. It's going to also explain and specify the position of your particular device within the therapeutic landscape. It will justify the choice of the criteria that you're using to demonstrate equivalence. It will help you define the criteria that you use for your risk-benefit analysis. And it will also help to define the context and the endpoints for your safety and performance assessments, which will include clinical investigations, your literature analysis, as well as your post-market surveillance. So on the next slide, we have just some general content that we use as a jumping off point. Each project will be very different, but generally speaking, it's expected that the state-of-the-art section will include a detailed description of the medical condition. So any of the particular clinical forms, stages, differing levels of severity. You'll also want to pull in information regarding the population that's affected and whether or not there are specific differences in the frequency or the appearance or clinical course of that particular condition, depending on the population. This is directly informed by your choice of PICO terms, so your population that you've identified. You'll also want to go into quite a bit of detail as far as the therapeutic management and diagnostic options. So those will include any available option or alternative, but also a description of any sort of historical context or the the development of the technology over the course of its existence. You should be prepared to explain the pros and cons of using different therapeutic or diagnostic options, and detail the benefit risk profiles and the acceptability. And then finally, you'll want to include a detailed description of the maturity of the technology. So how does your particular device perform? How does it fit within that therapeutic landscape? What are any hazards? And are there risk reduction approaches that can be taken? And basically just set up how your device seats within the entire landscape of all of these other alternative diagnostic or therapeutic devices. So if you think of the state-of-the-art section like a funnel, that's kind of the easiest analogy. You're going to start very broad. You're going to start with your medical condition or problem. You're going to describe that condition as well as any sort of clinical course and the different forms it may take, as well as provide some epidemiology if there are particular populations that are more directly affected. And this is where it's very, very similar between the state-of-the-art section within the MDR, as well as the SVR section within IVDR. Up until now, this is all very, very similar. Where it's going to start to diverge is in terms of your alternatives or the interventions. In IVDR, you are using an in vitro diagnostic device. So in terms of risk, it's going to be very different. The risk is going to be in terms of making a clinical diagnosis. What are the risks associated with making an incorrect diagnosis in terms of delaying treatment or initiating the improper treatment based on a wrong diagnosis? Whereas in MDR, if you have a treatment option, there's going to be that more direct impact associated with risk. But ultimately, the approach that you're going to take is going to be very similar in terms of that funnel, that increasing level of detail. You're going to describe the most conservative option for treatment or diagnosis. How would this, if your device or diagnostic tool was not available, what are the other options that a clinician could use to make that same treatment or diagnosis? So you would start very broadly, like let's say you have a device that's like a stent. You would start, if you're treating coronary artery disease, you would start very conservatively. You could have pharmacological interventions. You could try medicine, lifestyle changes, behavioral changes, that sort of thing. And then you get into more of the direct treatment, the interventional options. You'll go into details about the advantages and disadvantages of each of those different options. If you were using the same approach for IVDR, it would be similar in that there may be certain symptoms that would cause you to suspect a particular disease. That would be more the conservative. There's not a whole lot of conservative diagnoses. You want to have it be quite specific. So in that case, that first section, the conservative options are usually sort of a description of the symptoms that would lead you to suspect this particular illness. And then in terms of the interventional options, that's where you're going to, again, think about what would happen if your particular test was not available. How would you make that same diagnosis? Are there other similar tests available that maybe have different pros and cons, different levels of sensitivity or specificity? And then finally, you want to funnel down to the actual treatment portfolio, the actual landscape. Who are your closest competitors? Who are you going to compare yourself against as you're trying to determine what is considered to be the acceptability of performance and safety? So the lower level here is going to be defined by your landscape, your direct competitors. Whereas that middle section here, as far as the other options or alternatives, would be other technologies that are capable of treating the same disease or making the same diagnosis, but are not direct competitors of your particular device. And I think that's where we're hoping to go with this particular webinar today, is to go into more detail about how you can decide what goes into each of these different sections.
Great, thanks, Sarah. We've received so many questions leading up to this session today, so we will start to answer the ones that we've received so far. If at any point during this session you have a question for us, go ahead and click on the Q&A button on your Zoom menu bar and send us your questions, and we will get to them in the order that we receive them. So here we go with our first question. And Lori and Sarah, let me know when you can see it on your screen.
Okay, yep.
Yeah, we can see it.
Okay.
One thing I wanted to add before we get to this question, Olivia, is that the graphics that Sarah was showing, Olivia will put a link up to the white paper that those that we've developed, that those came from. If you're interested in the information and looking at more information from the white paper, I think Sarah participated in writing that white paper, and it might be of interest to the audience to download that off of our website.
Yep, I'll go ahead and put that link in the chat. You can go ahead and click on that link and download the full white paper.
Great,
All right.
great. Thanks everyone for your questions too. We're looking forward to answering them.
Okay, so this attendee is asking, at what stage of product development should the SOA, which is based on literature review, be established and written?
We get this question quite a bit. I think the most direct answer is early and often, with the understanding that the literature review may reveal some new potential issues that will have to be addressed in other documents. So for instance, let's say you have a product that's being used for a very specific indication, and you begin your literature review as part of your state-of-the-art analysis, and you're realizing that it's actually being used for off-label usage quite a bit. That could inform some of your other documents. Or let's say you have the reverse problem, where your device, your indications state that it can be used in a particular population, let's say a pediatric population. And then as you get into your literature review, you realize there is no literature in a pediatric population. That could also be an issue that would lead to the need for additional clinical studies as part of your own device's clinical performance. So really, as you're thinking about the state-of-the-art section, think about it more like a process rather than a product. This is going to be a continuing review of what is going on, getting a lay of the land as far as the options available to you. It's what's considered state of the art in terms of making a diagnosis or or treating a particular disease, but it's also based on the idea that technologies evolve and change. And as data is published, you want this to be sort of a revolving publication. Sometimes they may start to use a different scale for assessing the severity of a disease. You know, if there are updates to the scale as far as or staging of a particular disease, you may need to include that as part of your own assessment. You may find that certain performance measures are being included in more recent studies, or some of them that had been included historically are now not being used quite as often. That's really the purpose of the state of the art, is to help you inform the writing and the assembly of all of these other technical documentations, so that you can be consistent across all of them.
Okay, great. Thanks, Sarah. Moving on to our second question. This attendee is asking how to organize the flow of the state of the art.
So I think I'm hoping that white paper will do a really solid job of explaining our overall thinking. But again, picture that funnel. You want to start very broad. You want to think about all of the different ways of treating a particular disease. And then you want to get into very specifics. So you want to get down to your closest competitors. That will direct your choice of the data sets, the data that you're going to use to develop your acceptance criteria. So by organizing your state of the art to start very broadly and then move more and more specific and precise, you can ensure that your choice of competitors is supported by the rest of the document, and that it is organized in such a way that the notified body doesn't have to look for things. You want to make sure that these sections flow well together, that if you've defined a certain population that's being used, that it's carried through in terms of how does that population respond to different types of therapy therapeutics or different options. Maybe it works for one population, but not another. You want to make sure that you cross-reference and that you refer back to the different sections throughout your document. But usually that system of starting broad and moving more specific is the best approach.
And I've got some things to add on that. Thanks, Sarah. I noticed in Sarah's earlier slide where it kind of showed that funnel, there were some notes in that slide that said things like at the very top of the funnel where you're talking about medical conditions, a description of those, and it said off to the side for every relevant condition. So there were hints in there that that speak to the breadth of the state-of-the-art section in a CER. It is not uncommon for in our writing of CERs that the state-of-the-art section, of which Sarah is an expert at, are enormous sections. If your device, for example, has 5 different indications and medical conditions it treats or anatomic locations, you have to march through a description of all of those. Isn't that right, Sarah?
Yes, and make sure that those narratives are balanced. That's another big thing we've noticed, or you will have a giant background section on one indication, and then the next indication, you have basically a sentence. And the notified body is going to pick up on that. They should be treated with equal importance and supported with equal levels of evidence. Correct.
So these state-of-the-art sections can be, you should not give them short shrift. The notified bodies scrutinize them, in our experience, the notified bodies scrutinize them very heavily, and you start broad, you cover all your bases, and you work down the funnel through, the medical conditions. The therapeutic options, that second level of the funnel, can also be very broad. The watch and wait, the passive treatment options, pharmacologic treatment options, and then on the right side of that graphic were the interventional treatment options. That's usually where your device will live, and she described the difference between that and if you're talking about an IVD device. So the point that I wanted to make and build on Sarah's comments about this is that this is a large section. It's a complex section, and it's probably, in our estimation at Criterion Edge, it's the most important section of the CER. It sets up the rest of your CER for a good foundation to build on as you then turn your attention, CER or PER, after the state-of-the-art section, you turn your attention to the subject device. And so the SOA is a big section and should not be taken lightly. Sarah.
Okay, let's move on to our next question. Being new to the industry, what is the best approach to take when writing a CER?
I think the first advice I would give to you is to develop a checklist or start scoping early on. Figure out what you have available to you, what you don't have ready yet. If they are actively revising particular things like your IFU, those are important things to take note of very early on. Getting a timeline together early on. If you're expecting something to take 6 months, it's probably going to take 8. Always err on the side of caution. Know that this whole process, particularly if you have a device that sits in an area with a lot of very active research and a lot of publications, things like the systematic literature review can take quite a bit of time and man hours to go through all of those articles and extract all of that data. So I would say getting on a schedule, identifying what is available currently and what's outstanding, and then determining who is going to be responsible for pulling all of these articles together into one cohesive report. It's all about sort of like resourcing and organization. That's going to be critical.
Yeah, outlining and lining up your cross-functional team and everyone's job in that whole milieu of putting this clinical evaluation report. Because remember, a clinical evaluation report or performance evaluation report is just that. It is a report on the internal process of performance evaluation of the device or the clinical evaluation of the device. So it's a report on that. The CER or the PER shouldn't generate necessarily new information. It is a report on your process internally. So one big hint is, one big clue is, get your cross-functional team together, get everybody on the same page. Sarah mentioned the scoping of the report. That is a critical first step. And if you look no further than MedDev 2.7/1-4, where it talks about the first step in this report writing, this evaluation process, is scoping. So choose your leaders, choose who's responsible, give them the tools to get this done, and meet often to keep the team aligned.
Great, thanks, Lori. Moving right along. These two questions were submitted to us prior to our session today. What advice do you have for medical device benchmarking, and what is the best way to present the data in the state of the art if your device is the benchmark? Sarah, do you want to start us off on this one?
So we hear clients use the term benchmark quite a bit. So I just wanted to kind of define what I consider to be a benchmark. So for me, a benchmark device is similar to your competitors. It's a device that some other company makes with the same intended purpose, and that they're going to be most comparable in terms of their clinical performance and safety profile. So when you are developing a benchmark, when you're trying to decide who to benchmark, it's all about coming up with those closest competitors. Who is going to perform the most similar to you? So for instance, if again, with an MDR example, if you have a stent, are you going to include all stents or only drug-eluting stents? Are you going to include only one particular type of drug in the drug-eluting stents? How narrow do you want your focus to be? If you're trying to identify your closest competitors, usually you can get away with a handful. You don't want to have a massive number of competitors that you're going to search for. Usually like somewhere around 5 to 10. 10 would be on the very high end of that, but around 5 is usually pretty typical for us. It's usually those devices that, as you're doing your systematic literature review, as you're looking for comparisons in the literature between one device and another device, what tend to be the devices that most groups use is their sort of tried and true, their standard by which they would judge a new device on the market. So often you can get information about some of your closest competitors from your marketing division, but it's important that you also recognize any technical differences between your device and what you would consider to be a competitor, or, for benchmarking. And in the event that your device is the benchmark, if yours is the one that everyone else is striving to beat, that's totally fine. But you will want to present data on who claims to be your closest competitors in the literature and what are the similarities and differences in terms of the technology. Usually the best approach to this, I usually do a comparison table between our device and then all of the closest competitors that we've selected for the current state-of-the-art analysis. So I will include a comparison of my device, and then if there's a particular drug it uses, if it's indicated for certain sizes, sizes of vessel. If it's like if you're talking about IVDR, you could do something like, what is the actual target? If you're looking for something like a COVID test, are you detecting one particular, are you looking for the spike protein, or are you looking for the nucleocapsid protein? If they're both PCR tests, that's fine. You can include another PCR test, but if that particular PCR test uses a different primer set than yours, that's important to make note of, because that could lead to differences in your level of sensitivity or specificity for that particular test. It's all about identifying who is the absolute closest to you in terms of the technology. And that's going to
right. And Sarah, if I can jump in there on that, I think that the other issue at play here that we often hear, would you not agree, Sarah, that we often hear, but there is no other device like ours.
Right.
That does exactly what our device does. So who is our benchmark? Who are our competitors? And the approach that we've taken, and Sarah, you can expand maybe on this too, the approach that we've taken in that case is that it's not a perfect approach, but it seems to be accepted by the notified bodies when we have taken it. Is it, if your device has 10 different attributes, size, indication, whatever the attributes are, clinical, technical, or biological attributes, you may have a host of competitive devices that only share 1 or 2 or 3 of those attributes. And then you're comparing to, you can't compare to the entire device, but you can compare 4 of your device's attributes against 4 of another device's attributes. In other words, you're kind of taking multiple competitors and comparing parts of your device to those closest competitors. Is that something that you've experienced, Sarah?
Yes. Sometimes there isn't an exact competitor. Nobody's going to be exactly aligned with you, but that's where the discussion that follows and that direct comparison that previews the data set that they're about to see really come into play. You want to explain
and justification justification. Mm-hmm.
Yeah, you want to explain why you've selected them, any limitations in the competitors that you have identified. And then if you do see differences in terms of the performance objectives and the ranges that you've identified, be prepared to explain that your device offers this particular advantage over what you've identified as your competitors. Or it may have more of a limited scope in terms of, maybe your competitors are indicated for a broader range of indications. If you have a particular device that your competitor is used for all sorts of surgeries and yours is only indicated for certain types of surgeries, making sure you're limiting your data set to only the surgeries that your device is indicated for. We've seen that quite a bit too, because some surgeries, and even breaking it down by anatomic site, sometimes the safety objectives are very different for one vessel versus another. Having different ranges that you've calculated based on large vessels versus small vessels, or patients with certain comorbidities, that's going to be where it really comes down to explaining and justifying your choices.
Yes, this is a very great questions. We've gotten a few of those, even after our session today started. This is a struggle. There's no doubt about that. This is not easy to tackle. It requires some careful navigation, the CER or PER, careful and considered justifications, and a very clear story. We always say, as a notified body, the reviewer needs things clearly laid out for them. They don't want to see hypotheticals. They don't like to see, big leaps, or gaps in your reasoning or your justification. They want to see that you as a sponsor are recognizing gaps or recognizing perhaps shortfalls, and how do you justify those, or what is your approach to close those gaps? Wouldn't you agree with that too, Sarah?
Oh, definitely. You want to lay it out. They will see through it.
Yeah. Yes. Right. So great question. And again, we're assuming that benchmarking means kind of how we've described it today, is like the comparison of your device back to competitors, even if there's no one perfect competitor. That's, taking again emphasizing those clinical, technical, and biological. I know that's equivalence language. I understand that, but you can use those clinical, technical, and biological attributes of your subject device in the case of comparing it back to its competitors, so that you can, at the end of the day, establish your acceptance criteria for your safety and performance objectives. That's what we're talking about. So great set of questions. Thank you.
All right, we're getting closer to the end of our session here, so we'll keep running through as many more questions as we can. Here's our next one. How detailed does the acceptance criteria need to be based on the state of the art in similar devices?
So I think this is a good point to make is before I answer the question, is the difference between the performance measures and the acceptance criteria. The performance measure, safety and performance measures are going to be the things that you're measuring. So what is an outcome of using your particular device? It could be technical success. Like if your device, how often is, you can have procedural success, you can have technical success, you can have maybe there's a particular risk associated with that particular device, like a particular safety outcome. So, how often those are your performance measures. The acceptance criteria are going to be the ranges of the values that you obtain through your analysis of your closest competitors. So if you have, again, using the stent example, if you're looking at restenosis, so how often does the vessel experience restenosis? You may obtain a range anywhere from 3 to 5%. That 3 to 5% is your acceptance criteria. The outcome, the performance measure that you're including, is that restenosis. For some devices, you may keep it very, very broad. You may just say technical success, procedural success, and major adverse events. For other devices, we've seen them break it down by what type of adverse events. Are you talking about MI, and how do you define an MI? Are you talking about death? Are you talking about bleeding? What type of bleeding? You can make it as detailed as you'd like, but be prepared to justify why you're selecting those performance objectives. They don't need to see a list of every single possible performance objective ever. They want to see the ones that are most relevant to this type of device, and also the ones that tend to be called out within the clinical practice guidelines. So referring to your clinical practice guidelines to see what they often report as they're comparing one technique to another is a really good place to start. As you're trying to decide sort of what are the safety and performance measures that I want to capture, and how many of these ranges do I need for my particular device. And in terms of state-of-the-art and similar or competitor devices, you will want to explain how other options, all these other alternatives perform compared to yours. If the rates of major adverse events are 5% with other alternatives and only 4% with yours, or 2% with yours, your competitors may give you that 2% range, but the alternatives give you the 5% range. You wouldn't want to set your bar based on your alternatives, because they're not similar enough to your device. So it will look like you're sort of setting the bar real low if you compare it to those state-of-the-art alternatives. Or you could accidentally set it too high. If you're comparing a PCR COVID test to an antigen test, the sensitivity for a PCR test is way higher than an antigen test. You wouldn't want to set your bar, your acceptance criteria, based on a PCR test if you're an antigen test. You'll never meet that bar. So this is where it's really important to clearly define your alternatives and competitors, but also explain those overall ranges of what you've identified. The level of detail is going to be far higher for your competitors compared to the state of the art. We usually limit the analysis for the state of the art, the alternatives, to things like meta-analyses or systematic literature reviews, something that's going to provide you a high level overview of, comparing drug-eluting stents to drug-coated balloons. What is the range of the different outcomes? You can use that in like a meta-analysis that you can cite. But then as you're getting into your competitors, your drug-eluting stent versus other competitor drug-eluting stents, that's where you're going to go article by article by article and describe what range you've identified based on the current searches.
Right. So I do want to emphasize what you were just saying, Sarah, is that acceptance criteria of your safety and performance, your identified safety and performance objectives of your subject device, the acceptance criteria are pulled directly from literature on the competitors. It's not registry data pulled from guidelines. It's pulled from because you have now established who the direct competitors are of your device. And so the notified bodies are interested in, well, how are the direct competitors of your device performing? And that your device's data must be at least as good as the direct competitors established within the state-of-the-art landscape. That's the most important part of that sentence, is that's why this whole process exists in the state-of-the-art section. Because at the end of the day, this is about comparing your device against other state-of-the-art competitors for the same, in the same patient population, same therapeutic option, and so forth. So that's why the context is important. Wouldn't you agree, Sarah? That's the crux of all this, is that that's what MDR and IVDR demand, is that how does your device compare to the state of the art of other devices similar or competitive to yours, correct?
Right, exactly.
Yeah. Yes. So it can't be the data coming from registries or more meta-analyses or what pooled data. It has to be against those direct competitors.
Okay. Moving on to our next question. What strategy do you think would work best for legacy, wet, or low-risk devices?
So this is another issue that we come up with quite a bit. I think the important thing to remember is that you cannot just expect them to accept the fact that you are a legacy or accept the fact that you're well-established technology. You have to meet the criteria and establish that early in your state-of-the-art section. So you want to justify why you may be considered a well-established technology, why you would be considered a legacy. In terms of risk, that's again where comparing to the alternatives will be extremely important. So you will still be held to the same rigor as far as identifying your competitors, looking for how well you perform compared to those competitors, but you will want to really point out why the risk level is what you say it to be. You want to make sure that that's supported by the data or supported by clinical practice guidelines. And in terms of the well-established technology, often that we come across that quite a bit. I think that goes back to looking at some of the longer-term meta-analyses or guidelines that will show the long-term use of a particular device for a specific indication. So you will, while your current state-of-the-art search is limited to the past 5 years, typically for a well-established technology, you may refer to some of the more systematic reviews and things that do go into more of that historic context.
Great, thanks, Sarah. Here's our next question. This attendee would like to know the purpose of putting similar devices in the state-of-the-art section.
So I think it depends on your definition of similar, and this would be something that I would definitely caution you against using that word in the state of the art because it can get confusing. We typically prefer to use a competitor or an alternative. So an alternative device, you would include those in the state of the art section, basically to help you establish that funnel, to show that if your particular technology is not indicated, or if your technology has evolved out of an unmet need that the other technologies cannot meet. That's really the purpose behind keeping those other tests in there. And it could be that your particular device is not the most technically advanced, the newest thing on the market. It is one of those legacy devices where, yes, there are more efficient drugs on the market or more efficient tests on the market, but your device still serves a very specific purpose under a specific set of circumstances. So really, by including those alternatives in your discussion, you help to set the stage of under what conditions would your device be considered for use, and when is it actually preferred for use, under what circumstances.
So Sarah, you and I have discussed before this use of the word similar devices. And I think you spoke to it at the beginning of your answer, is that, when Sarah and I see these questions, we don't use the term similar in any of our CERs because we're not really sure what it means. We use equivalent, we use competitor, but we don't use the word similar. So equivalent is what that says it is that we have established that the company, the manufacturer, holds another device in which equivalence can be established between that device and the subject device of the clinical evaluation, for example. That's equivalence. That has a big multiple bars to cross to establish equivalence between two devices that are very, very exacting. So they're equivalent devices, they're not similar. And then there's a competitor, which I guess are similar, but they're competitor devices. You establish them as that. So we're never quite sure what similar means to different people. And maybe we're behind the times. Maybe that's a term that's being used in CERs. We just have never really seen it in CERs, and we don't really know how to use it. So that's a little bit of our disclaimer about the use of the word similar. It feels like a vague, if you will, catch-all term, and we're not quite sure how to approach the use of the term similar devices. Is that what you think too, Sarah?
Yeah. I would just say this is a really important decision to make very early on too, to make sure that you understand sort of who's your competitor versus who's your alternative.
Right. Is a similar device an alternative device? Then call it an alternative device. Is a similar device an equivalent device? Well, that's a whole another animal and goes in a whole section by itself in the CER. Or is a similar device the competitor device? In which case, call it the competitor device. That's kind of where we land in the use of the word similar.
Mm-hmm.
All right. Thanks, Lori, and thank you, Sarah. I think that's a good place to end on. We are out of time. I hope that Sarah and Lori were able to answer most of your questions, but if we didn't get to yours, we'll follow up with you later today by email. Thank you again, Sarah and Lori and everyone for joining us. Take care, and I hope to see you at our next webinar. Have a great day.